Hello, thank you for having me. Today I'll be talking about the spatial organization of bacterial genetic diversity in the gut microbiome. So briefly, we observe an immense amount of genetic diversity in the gut microbiome, which really comes from two sources. One is from the co-colonization of genetically distinct strains of the same species. So within a given host, we often see between 1 and 4 of these genetically distinct lineages that are stable over long time periods. Here I'm showing a host with 3 strains stable over a year. And these genetically distinct lineages tend to be diverged by about 10,000 single nucleotide variants, or SNVs. So that's an immense amount of genetic diversity. In addition, on individual strains, we see genetic variation occurring through within-host evolution. So if we were to focus on one population, say the teal population of bacteria, we might see individual nucleotides actually arise through mutation and rise to intermediate or high frequency over time. And we really have observed both of these processes through fecal sampling, which is a really powerful method because it, one, is non-invasive, and two, has a huge amount of bacterial biomass, allowing us to deeply sequence samples in order to achieve these kind of genetic or evolutionary analyses. But it potentially allows important spatial organization that might exist along the gastrointestinal tract. So that leads to kind of the main question of my study is bacterial genetic diversity spatially distributed? We do see spatial organization at the level of species. So for example, oxygen gradients in the gut maintain facultative anaerobic taxa in the upper gut, obligate anaerobic taxa in the lower gut. And while this environmental heterogeneity maintains species diversity, we don't really know if it does the same at the genetic level. So I'm kind of testing 2 alternative models, one in which bacterial genetic diversity, such as strains, spatially segregate into distinct niches, for example, to reduce competition, or alternatively, if, for example, co-colonizing strains are actually well mixed along the gut, implying that some kind of other ecological force other than spatial segregation is maintaining strain diversity. Considering mutations, we might ask, do these mutations sweep only within particular physical niches, or are they global? Do we find them sweeping throughout the gut environment in accordance with a well-mixed model? So to answer these questions, I leveraged a data set provided to us by our collaborators at University of British Columbia in the lab of Dr. Carolina Tropini. So they took 8 gnotobiotic mice and inoculated them with the same human stool-derived inoculum, so the same collection of species and strains. These mice were allowed to equilibrate for 8 weeks before we collected luminal samples from 5 sites along the gut, shotgun sequenced them, and mapped the short reads to a reference database of bacterial genomes, allowing us to call, for example, species abundance, but also the abundance of individual genetic variants. And with this data set, I set out to answer 3 questions: the spatial distribution of species, of strains, and evolutionary changes. So I'll start with the species question. Now, as I said, we already expect to see spatial heterogeneity and species abundance. And we mostly wanted to see that in humanizing mice, we were recapitulating this pattern. And briefly, here's a plot of relative abundance of taxa along the gut. And we do see that there is spatial heterogeneity, as we would expect. In particular, these obligate anaerobic taxa, such as Bacteroidales, is expanding in the cecum and colon, recapitulating kind of the expected patterns of species-level diversity. But what about at the subspecific level? For example, at the level of the strain? I'll make a brief methodological point. It's no simple thing to infer strain frequency from short-read data. We did so in a similar way that my lab did so in a previous paper, whereby we leveraged linkage disequilibrium displayed by SNVs that travel on the same genomic or strain backbone. I can get more into this in the question and answer section, but briefly, we looked for these clusters of SNVs showing LD across my mouse samples, which are shown along the x-axis, so from mouse 1 to 8 on the x-axis, and then the frequency of individual SNVs or strains are shown on the y-axis. The gray lines are individual SNVs. So we essentially isolated SNVs that seem to be traveling on the same strain background, allowing us to assess changes in strain frequency across samples, including within hosts. So I'm going to show the same plot in a slightly reformatted plot that's easy for us microbiome scientists who are used to these stacked bar plots to read. So here I'm showing a case study of a species called Blauschia wexlerae. This study had 2 strains in the inoculum, and we can observe how those 2 strains ultimately colonize our 8 mice, shown from left to right. And we can see that strain frequency or colonization dynamics are quite variable between mice. But when we zoom in on a single mouse, we can see that actually a story of spatial uniformity emerges, that even when 2 strains colonize, they tend to do so at similar frequencies all along the gut. And this pattern of spatial uniformity is something that we saw with all of the species to which we could apply this analysis, which had multiple co-colonizing strains that got into our mouse cohort, which implies that strain stability through time and space is facilitated through niche partitioning. For example, metabolic niche partitioning, as opposed to this kind of spatial segregation model that I had originally proposed. Now, what about below the level of the strain to individual genetic variants? So here I tracked SNV changes as like a single nucleotide variant that went from low frequency in one sample, say location 1, to high frequency in another sample, as long as those 2 samples harbored the same dominant strain. And we can ask, do these SNVs tend to evolutionarily differentiate between different sites in the same hosts, so spatially segregated, or do they not? So here I'm going to focus on another case study that kind of reveals the broad scale patterns that ultimately emerged. Here I'm looking at a species, population of a species called Pedis dissonus, and we're going to look at a single SNV in this starch utilization gene in the SUSC gene family. And this SNV went from low frequency in the inoculum to high frequency, actually in multiple independent cages. And this kind of pattern of independent replication actually is a hallmark of adaptation. We call it parallel evolution or evolutionary parallelism. But actually the pattern I want to draw your attention to is the fact that when it sweeps to high frequency, it does so all along the gut. And in fact, even when it sweeps to intermediate frequencies in these remaining 3 mice, it's doing so at relatively uniform frequencies all along the GI tract. So we asked if this pattern were in fact generalizable to all the evolutionary changes we observed. We detected 65 evolutionary changes across 30 species, and we aggregated this into a single evolutionary change rate or SNV change rate. And we find that evolution is occurring. For example, we detect SNV changes between the inoculum and our mouse hosts. But when we look within mouse hosts and ask, are there evolutionary differentiation along the GI tract, we see that it's close to 0. There's almost no kind of spatial differentiation within hosts. So this brings me to my conclusions. One, species are display heterogeneous frequencies along the GI tract, as we expect. But contrastingly, strains have uniform frequencies along the gut, indicating that some other ecological force besides spatial segregation is actually permitting these co-colonizing strains to coexist within the same hosts. And finally, when going to the level of individual genetic variants within a strain population, we see that these two rise to high frequency throughout the GI tract. They're not spatially specific, indicating that there's either weak local selection pressures or else rapid migration that's actually homogenizing the landscape of genetic variation all up and down the GI tract. So in future work, we'd like to explore this in other contexts, such as natural human gut microbiome, the natural mouse microbiome, or in the context of pathogenesis and disease. With that, I'd like to acknowledge my funding source, my PI, my colleagues in the Garuda Trapani Lab, and my committee members. Thank you.